Okay, so think outside the box. Process in science often depends on people thinking outside the box. Original thinkers who are often ignored or even ridiculed when they first put forward the new ideas. One such individual who battled constantly throughout her career to get her ideas accepted was the American biologist Lynn Margulis. Her greatest achievement was to use evidence from microbiology to help firmly establish an idea that had been around since the mid-19th century, that new organisms can be created from combinations of existing organisms which are not necessarily closely related. The organisms form a symbiotic partnership, typically by one engulfing the other. A process known as endosymbiosis, dramatic evolutionary changes result. The classic example, now confirmed by later work, are the suggestions that mitochondria and chloroplasts are originally free-living bacteria, prokaryotes, which invaded the ancestors of modern eukaryotic cells. Margulis saw such symbiotic unions as a major driving cause of evolutionary change. She continued to challenge the Darwinian view that evolution occurs mainly as a result of competition between species. So yeah, this is basically a motivation for you to how a scientist approaches, like, with confidence. So Darwin gave the idea that evolution occurs mainly because of competition, but this scientist has given the idea, or is like trying to stand the idea, that also symbiotic relation can also cause an evolutionary change. Okay, so in the early days of my microscopy, an English scientist, Robert Hooke, decided to examine thin slices of plant material. He chose cork as one of his examples. So cork is basically, a mature cork cell is basically a dead cell which is found inside the trees. Yeah. So looking down the microscope, he was struck by the regular appearance of the structure, and in 1965 he wrote a book containing the diagram shown in figure 120. So this is the diagram this is the picture that he saw while he was observing the cork cell, cork tissues. Actually, it was not a cell, it was a tissue because it was a part under the microscope. Yeah. So if you examine the diagram, you will see the pore-like regular structures that Hooke called cells. Each cell appeared to be an empty box surrounded by a wall. Hooke had discovered and described without revealing it the fundamental unit of all living things, which is the cell. So why is this the fundamental living unit? What is a unit? A unit is like a group of things like combined together to form something that does something. So basically cells are a group of organelles together with cytoplasm that does the basic function of a living thing. Okay, although we know that the cells of cork are dead, further observations of cells in living materials were made by Hooke and other scientists. However, it was not until almost 200 years later that a general cell theory emerged from the work of the two German scientists. In 1938, Schleiden, a botanist, suggested that all plants are made of cells, and a year later Schwann, a zoologist, suggested the same for animals. So plants and animals are made of cells. It is the basic fundamental unit of a life. So it was further clarified by further researches. Okay, the cell theory states that the basic unit of structure and function of all living organisms is the cell. Now, over 170 years later, this idea is one of the most familiar and important theories in biology. So, like, why structure and function? Definitely, cells must have a structure and cells must have a function, like the liver cell, the kidney cells, or the heart cells, or the muscle cells, the nerve cells, the body cells, the germ cells. So these are the different types of cells that does a function, and a cell is, since it is a unit, it consists of few other things which are organelles and cytoplasm that works together. We'll see them in great detail later. Now, over 170 years later, this idea is one of the most familiar and important theories in biology. To it has been added Virchow's theory of 1855 that all cells arise from pre-existing cells by cell division. What does it mean? Well, it means that say for example this is a cell. This cell will divide by mitosis to form two cells. this cell will again divide to form other two cells, and this cell will again divide to form other two cells. Like this, cell division keeps going on and on and on. So like cells are made from the pre-existing cells. Yeah, this was also true, and it is happened because of mitotic cell division. There is another type of cell division which is meiotic cell division, which has half the chromosomes of its parent cell, because it happens in the germ cells, reproductive cells. We will look at that later. But why mitosis? We know that DNA is has all the instruction to control the cell. So all the daughter cells, I mean the cells produced from the original cell, the original cell is the father, and the cells produced are the daughter cells. So the daughter cells must have the same genetic materials and everything. That's why it must undergo mitotic cell division. We'll look at the different types of cell division in great details later. Okay. So nothing much else to explain here. Why cells? A cell can be thought of as a bag in which chemistry of life is allowed to occur, partially permeated, partially separated from the environment outside the cell. So think about this. This is the cell, and this is the, suppose that this is the environment in which the cell is. Okay, just for your understanding, it doesn't have to be a perfect drawing. And all the chemistry of life, it means that the chemical reactions which are needed for our life to survive, to grow, to keep moving forward, are happening inside the cells. Why? Because the environment, look here, the environment do not have the perfect conditions for the chemical reactions to occur. But inside the cell, since it is partially permeable by the cell membrane, only the chemicals that are needed for the chemical reactions and the environment which is needed are formed inside the cells. So that's why chemical reactions can occur, as it has a suitable environment. Thus, like a cell is basically a bag in which you can adjust the environment and the materials so that the chemistry of life needed for the life can take place easily. Yeah, that's basically what a cell is. Think of it this way for now. And it is partially separated from the outside the cell by the cell membrane. Why is it partially? Because it allows some chemicals to get in and some chemicals to get out. Now, which chemicals can get in and which chemicals can get out are decided by the cell, because it will understand that which chemicals it needs and which chemical it needs to get out of the cell. And how the cell membrane does this choosing and partially permeable transfer of materials, we will also learn it in great details in the later chapters. Transporter proteins are, we will learn it later. Okay, so for now this will be enough. So the thin membrane which surrounds the cell is essential in controlling exchanges between the cell and its environment. It is a very effective barrier, but also allows a controlled traffic of materials across it in both directions. The membrane is therefore described as partially permeable. If it were freely permeable, life could not exist, because the chemicals of cells would simply mix with the surrounding chemicals by diffusion, and so the surrounding chemicals like unwanted chemicals may enter the cell, and so that's not the environment that you want. And what is diffusion? We'll also learn that later. Diffusion is basically the movement of water molecules from higher water potential to lower water potential. Yeah, diffusion of that's osmosis, and diffusion of water is osmosis, and diffusion of other chemicals can happen. Like for example, we need chlorine inside the cell. If chlorine is higher in the outside environment, it can simply come inside the cell by diffusion, which is the cell has lower concentration. And can the chlorine come in the cell membrane if it allows? So a lot of chemical reactions occurring, we will see them. This is the first chapter, so it is very basic chapter. So cell biology and microscope. The study of cells has given rise to an important branch of biology known as cell biology. Cells can now be studied by many different methods, but scientists began simply by looking at them using various types of microscopes. There are two fundamentally different types of microscope now in use: the light microscope and the electron microscope. Both use a form of radiation in order to create an image of the specimen being examined. The light microscope uses light as a source of radiation, while the electron microscope uses electron for reasons which are discussed later. So we cannot see the cells and the organelles inside the cells. Oh, by the organelles are like the other things, small organs, you can say, inside the cell, like the mitochondria, nucleus. Yeah, we will look at that later, but we need a microscope, and there are two types of microscope. So first let us see what's a light microscope. So basically this is a light microscope which uses light to look at the specimen. We just need to understand, know the parts of the microscope and how to use it, but not how it works, because that's the work of the students of physics. They will know. We, as students of biology, we do not need to know how the microscope works. We just need to know how to make it work so that we can use it to examine the cells and chemical reactions, because we are biologists. Yeah. So the golden age of light microscopy could be said to be the 19th century. Microscopes had been available since the beginning of the 17th century, but when dramatic improvements are made in the quality of glassness in the early 19th century, interest among scientists became widespread. The fascination of the microscopic world that opened up in biology inspired rapid progress both in microscope design and, equally importantly, in preparing material for examination with microscope, because you cannot just take a material and examine it under the microscope. You have to first prepare it. We will look at that later. This branch of biology is known as cytology, like examination of the cells. It's called cytology. Figure 1.3 shows how light microscope works. By 1900, all structures shown in figure 1.4 and 1.5 had been discovered. So figure 1.4 is the structure of a cell of an animal, and figure 1.5 of a plant. They have differences and similarities. Let me first show you the structure of animal. So the animal cell has mitochondria. These organelles are called the mitochondria. They are the powers of the cell. They does the chemical reactions which produces energy for the cells in the form of ATP and aerobic respiration. Aerobic respiration is like the process. Like aerobic respiration is the respiration in which we use oxygen to make the energy of the cell, and it is done in the mitochondria, and the energy is in the form of ATP. We'll learn it in great detail later. Cell membrane, I have explained cell membrane. So nucleus is basically the brain of the cell, which coordinates how the cell's chemical reactions are going to work. It has the DNA, which instructs the cell which proteins should be made, and proteins does the function. Now let's see the nucleus also has a membrane which is called the nuclear envelope. It also is partially permeable and allows the entry and exit of the chemicals which the nucleus wants. There is chromatin. Chromatin is basically groups of nucleotides form a DNA. Groups of DNA forms genes. Groups of genes forms chromatins. So chromatin is basically long thin fibers of DNAs or genes. Okay, so this DNA has the instructions or the codes of how to make the which protein, and protein is made by ribosomes, another organelle, and ribosomes are made by the nucleolus. The nucleolus makes the ribosomal RNA, which forms the ribosomes. So yeah, ribosomes is another organelle and it is produced by the nucleolus. It is not directly produced, like nucleolus produces the ribosomal RNA, which produces the ribosomes, and thus with the help of like nucleus, messenger RNA, transfer RNA, ribosomal RNA, ribosomes, protein is made. We will see them in great details in the later chapter. Centriole is always found near the nucleus, has a role in nuclear division. Well, in nuclear division, we need the centrioles. Basically, the centrioles pulls. We will know that in great detail, but let me just tell you in short that centrioles pulls the chromatids from the chromosome. So, during cell division, the chromatins forms into chromosomes, and each organism has a specific set of chromosomes which are its identity. We will learn that later. Cytoplasm, yes, we know what is the cytoplasm. It is where all the chemical reaction occurs where all the materials are present. Golgi body. Golgi body is produced from the endoplasmic reticulum. There are two types of endoplasmic reticulum: smooth endoplasmic reticulum and rough endoplasmic reticulum. Rough endoplasmic reticulum contains ribosomes. Ribosomes are also free in the cell. So rough endoplasmic reticulum's basic function is to produce the proteins, and smooth endoplasmic reticulum produces the fats and steroids. It also makes the Golgi body. The Golgi body's function is to process the proteins produced by the ribosomes and to package them. Now let me tell you that the small endoplasmic reticulum's proteins are broken down and Golgi bodies are formed. Yeah, so that's it. This is animal cell, and plant cell has similarities and differences. It has chloroplast, which contains the chlorophylls. Chlorophylls are present in the grana of the chloroplast, where photosynthesis occurs to make the carbohydrate, because we know that plant cells can make their own food, but animal cells cannot make their own food. And cell wall also have plant cells also have cell wall, which is totally permeable, but it is needed for protection and for rigidity. And cell wall has middle lamella, like holding cells together, contains calcium pectate. Nothing to make you understand. There is plasmodesmata, which connects the near-bearing cytoplasm of the cells. Yes, there is also cell membrane, because cell membrane controls the exit and entry of the nutrients, but there is still plasmodesmata. And in plant cells there is a large vacuole. In animal cells there are small vacuoles, okay? But in plant cells there are large vacuoles. So the vacuoles is another membrane bond structure, which contains few chemicals, and these are separated from the cytoplasm. So yeah, nothing much to explain. There are a few differences and similarities. We will look at them right now. Animal and plant cells have features in common. In animal cells and plant cells, each cell is surrounded by very thin surface membrane. This is also sometimes referred to as plasma membrane. Yes, I told you. Many of the cells' contents are colorless and transparent, so they need to be stained to be seen. Each cell has a nucleus, which is relatively large structure that stains intensely and is therefore very conspicuous. The deeply staining material in the nucleus is called chromatin and is a mass of loosely coiled threads. This material collects together to form visible separate chromosomes during nuclear division. It contains DNA, deoxyribonucleic acid, and RNA is like ribonucleic acid. So RNA contains one extra oxygen, and we will also look at the structure of DNA and RNA in great detail later. So yeah, DNA is basically the codes that produces the protein. So yeah, we will learn that in great detail, but this is just a basic understanding. A molecule which contains the instructions that control the activities of the cell. Within the nucleus and even more deeply staining area is visible the nucleolus, which is made of loops of DNA from several chromosomes. The number of nucleoli is varied, 1 to 5 being common in mammals. The material between the nucleus and the cell surface membrane is known as cytoplasm. Yeah, cytoplasm is an aqueous watery material varying from a fluid to a jelly-like consistency. Many small structures can be seen within. These are often likened to small organs and hence known as organelles. So those organelles has different functions. They do different functions. And organelles can be defined as functionally and structurally distinct part of a cell. So different organelles have different structure and different functions. Organelles themselves are often surrounded by membranes so that their activities can be separated from the surrounding cytoplasm. This describes compartmentalization, because each organelle's chemical reactions are separated from the cytoplasm and other organelles and the cells. So there are different compartments. Having separate compartments is essential for a structure as complex as an animal or plant cell to work efficiently. Since each type of organelle has its own function, the cell has said to show division of labor, sharing the work between different specialized organelles. So division of labor is basically work, and the work is divided across different organelles. So that's why there is a division of labor. It's not like that one cell or one organelle is doing all of the work. The work is divided, like energy creation is done by the mitochondria, protein creation by ribosomes, and sorting and processing the proteins by the Golgi apparatus. Yeah, so the most numerous organelles seen with the light microscope are usually mitochondria. Mitochondria are only just visible, but films of living cells taken with the aid of a light microscope have shown that they can move about, change shape, and divide. They are specialized to carry out aerobic respiration. The use of specialized stains containing silver enabled the Golgi apparatus to be detected for the first time in 1998 by Camillo Golgi. The Golgi apparatus is the part of a complex internal sorting and distribution system within the cell. It also sometimes called Golgi body or Golgi apparatus. I have explained the function of Golgi body. So yeah, now let us see the differences between animals and plant cells. The only structure commonly found in animal cells which is absent from plant cell is centriole. Well, centriole has the function in cell division, but I guess plants do not need them. We will see the function of centriole in plant division, animal cell division, both in mitosis and meiosis. And while cells divide, the genetic materials need to be divided equally in mitotic cell division, which occurs in somatic cells, and 50-50 while it occurs in meiotic cell division. Plant cells also differ from animal cells in possessing cell walls, large permanent vacuoles, and chloroplasts. Okay. Centrioles. Under the light microscope, centrioles appears as a small structure close to the nucleus. Centrioles are discussed on page 18. Cell walls and plasmodesmata. With the light microscope, individual plant cells are more easily seen than animal cells because they are usually larger, and unlike animal cells surrounded by cell wall outside the cell surface membrane, this is relatively rigid because it contains fibers of cellulose, a polysaccharide which strengthens the wall. Rigid means it cannot be if pressure is exerted, it won't change its shape because it is a cell wall which is very rigid, and I told you that it's totally permeable. So the function is actually to protect and the rigidity because plants may have to go through harsh environment. The cell wall gives the cell a definite shape. Oh, and what is it contains fibers of cellulose, but what is a polysaccharide? We will learn this in the later chapters. The cell wall gives the cell a definite shape. It prevents the cell from bursting when water enters by osmosis, allowing large pressures to develop inside the cell, because suppose the water is just constantly diffusing into the cell has gone bigger and bigger, and poof, it will burst. That is if it's an animal cell. But if it has something harder apart from the cell membrane, very hard, so that hard part won't be stretched so much that it will burst. So it also protects the plants. And we will learn that osmosis occurs in animal cells to control the water potential and other temperature and other environment of the cell. But in plants, osmosis do not occur. So that's why they need to have extra protection. In animals, if extra water keeps coming into the cell, then osmosis will occur to correct that mistake. We will see that later. Cell walls may also be reinforced with extra cellulose or with a hard material called lignin for extra strength. So extra cellulose may be also added into the cell wall, or a hard material called lignin can be added for extra strength. Cell walls are freely permeable, allowing free movement of molecules and ions through the cell surface membrane. Plant cells are linked to neighboring cells by means of fine strands of cytoplasm called plasmodesmata, which pass through pore-like structures in their walls. Movement through pore is thought to be controlled by the structure of the pores. Nothing much to explain here. Vacuoles. Although animal cells may possess small vacuoles such as phagocytic vacuoles. So phagocytic vacuoles is small vacuoles produced inside the cell because of the process of phagocytosis. Phagocytosis is the process by which our white blood cells engulf the bacteria. We will see that later, but they are not permanent. They are temporary and small, which are temporary structures. Mature plant cells often possess a large permanent central vacuole. Yeah, the large permanent central vacuole. The plant vacuole is surrounded by a membrane, the tonoplast. The Membrane of the cell is called cell membrane. The membrane of the nucleus is called nuclear envelope. The membrane of the cell vacuole is called tonoplast, which controls exchange between the vacuole and the cytoplasm. And we know that membranes can control the exchange of materials between its environment. The fluid in the vacuole is a solution of pigments, enzymes, sugars, and other organic compounds, including some waste products, mineral salts, oxygen, and carbon dioxide. So, what are pigments? Pigments are basically the colored part of the material, chemicals in the cell that makes that organ colored. Yeah, so it contains, these are good information. Vacuoles have to regulate osmotic properties of cells, the flow of water inwards and outwards, as well as having a wide range of other functions. How does it control the osmotic properties of a cell? If the vacuole has a lot of water, the water potential inside the cell is increased. If the vacuole have less water, then the water potential in the cell has been decreased. So it can control the osmotic properties. And how is that? Does that? We'll also look at that later. There are transporter proteins called aquaporins, which transports water across the cells. Yeah. And there are like two types of transportation. One is the active transport, another the passive transport. Active transport occurs from lower concentration to higher concentration. Passive transport occurs from higher concentration to lower concentration. Which transport is needed and which chemicals should be transported is decided by the cell, and there are specific transporter proteins in the cells for doing that transporting function. Okay, we'll look at that in the later chapters. Vacuoles help to regulate the osmotic properties of cell. For example, the pigments which color the petals of certain flowers and parts of some vegetables, such as the red pigment of beetroot, may be located in the vacuoles. Good. Now, chloroplasts are found in green parts of the plants, mainly in the leaves. They are relatively large organelles and so are easily seen with light microscope. It is even possible to see tiny grains of or grana inside the chloroplasts using the light microscope. So the grana contains the chlorophyll, which traps the sunlight. Sunlight is needed for photosynthesis, which produces the food for the plants. When we know that animals cannot make their own food, that's why they do not have chlorophyll. They cannot make their own food because they do not have chlorophyll or chloroplast. So we'll look at this process in the great detail in the later chapters. The green pigment, these are parts of the chloroplasts. The grana are the parts of the chloroplast that contain chlorophyll, the green pigment which absorbs light during the process of photosynthesis. The main function of chloroplasts. So the main function of chloroplasts is to perform the process of photosynthesis. Chloroplasts are discussed further in page 19. Points to note: you can think of a plant cell as being very similar to an animal cell but with extra structures. So you know that there are differences and similarities we have just studied, and also we have also studied the functions of those organelles which cause the difference in both the cells. We'll learn them in great detail in the later chapters, of course. Plant cells are often larger than animal cells, although cell size is varies enormously. Do not confuse the cell wall with the cell surface membrane. Cell walls are relatively thick and physically strong, while cell surface membrane are very thin. Cell walls are freely permeable, whereas cell surface membranes are partially permeable. All cells have a cell surface membrane, but only plant cells have cell walls. Nothing much to explain. I have already explained this. Vacuoles are not confined to plant cells. Animal cells may have small vacuoles, such as phagocytotic vacuoles, although these are usually not permanent structure. It's not like that only plant cells have a big permanent vacuole. Animal cells may have small temporary vacuoles caused by many reactions like phagocytosis. We will look at phagocytosis in the later chapter in immune response. Okay, so return to the differences between animal and plant cells as seen using the light microscope on page 13. Okay, so units of measurement. In order to measure objects in the microscopic world, we need to use very small units of measurement which are unfamiliar to most of the people. According to the international agreement, the International System of Units, SI units, should be used. In this system, the basic unit of length is meter. Additional units can also be created in multiples of thousands times larger or smaller using standard prefixes. For example, the prefix kilo means 1000 times, thus 1 kilometer is 1000 meters. The units of length relevant to cell studies are shown in figure table 1.1. Yeah, we can see that these are basically the units, nanometers and millimeters. Nothing to explain, it's normal maths. It is difficult to imagine how small these units are, but when looking down a microscope and seeing cells clearly, we should not forget how amazingly small the cells actually are. The smallest structure visible with the human eye is about 50 to 100 micrometer in diameter. Your body contains about 60 million cells, varying in size from about 5 micrometer to 40 micrometers. Yeah, so nothing to explain here. Try to imagine structures like mitochondria, which have an average of diameter of 1 micrometer, the smallest cell organelles we deal with in this book. The smallest cell organelles we deal in this book, ribosomes, are only 25 nanometer in diameter. You could line up 20,000 micro ribosomes across the full stop at the end of this sentence. So it's very small. You can see here that, yeah. But now what is magnification? Magnification is the number of times larger an image is than the actual size of the object. So the formula of magnification is magnification equals to observed size of the image by actual size. So if we know the magnification, which is basically known because we know how much times you are magnifying by it is written on the lens, and if we observe the size of the image, like what is the size of the image by using the microscope, then we can calculate the actual size. Very easy. Here I equals to observed size of the image, that is what you can measure with a ruler, and A equals to actual size, that is the real size. For example, the size of a cell before it is magnified. If you know two of these values, you can work out the third one. For example, it is if the observed size of the image and the magnification are known, you can work out the actual size. Yeah, if you write the formula in a triangle. So basically nothing much to explain here. As shown in the right and cover of the values you want to find, it should be obvious to do the right calculation. Some work examples are not provided. This triangle is quite difficult. Don't look at this, just look at this. Put the value of magnification in M, the value of observed size of the image in I, and then simply calculate the A.